Good afternoon, everyone. Today, I would like to talk about the 2002 Nobel Prize in Physics. I chose this topic because it shows how scientists can discover invisible things and greatly expand our understanding of the universe. The prize was awarded for pioneering contributions to astrophysics, especially for the detection of cosmic neutrinos and for the discovery of cosmic X-ray sources. At first, this may sound difficult, but the basic idea is very interesting. The universe contains many objects and events that cannot be studied with ordinary visible light alone. Some of the most important information comes from signals that are extremely hard to detect. One example is the neutrino. A neutrino is an extremely small particle that interacts only very weakly with matter. Because of this, neutrinos can pass through stars, planets, and even our bodies almost without being stopped. This makes them very difficult to detect, but also very valuable for science. They can carry direct information from deep inside the sun and from violent events in space. To detect them, scientists build very large underground detectors. When a neutrino occasionally interacts with matter, it produces a tiny signal that can be observed. Another important breakthrough involved X-rays from space. Unlike visible light, X-rays reveal extremely energetic processes such as those connected with neutron stars and black holes. By detecting cosmic X-ray sources, scientists were able to study some of the most extreme regions in the universe. This prize was very important. It showed that the universe can be explored not only through ordinary light, but also through much more elusive signals. It also opened new ways to understand how stars evolve and how matter behaves under extreme conditions. I find this discovery fascinating. It shows that reality includes many important things that are completely invisible to our eyes. It also teaches us that progress in science often depends on creating new ways to observe what seemed impossible to detect. In the future, research on neutrinos and cosmic X-rays may help us understand the origin of the universe and the behavior of its most powerful objects. Even though this topic may seem abstract, it gives us an important message. The universe becomes richer and clearer when we learn how to detect what is hidden. Thank you.
Good afternoon, everyone. Today, I would like to talk about the 2002 Nobel Prize in Chemistry. I chose this topic because it shows how science can reveal the structure of life by making invisible molecules easier to study. The prize was awarded for the development of methods for the identification and structure analysis of biological macromolecules. At first, this may sound technical, but the main idea is quite easy to understand. Our bodies are made of many important molecules, such as proteins. These molecules are essential for life, but they are extremely small and very difficult to observe directly. Scientists wanted to answer two important questions. First, what kind of molecule is this? Second, what shape does it have? These questions are very important because the function of a biological molecule often depends on its structure. One major breakthrough came from mass spectrometry. This method made it possible to measure large biological molecules more accurately than before. A key breakthrough was soft desorption ionization, which allowed fragile biological macromolecules to be turned into ions and analyzed without being broken apart. In simple terms, scientists could finally identify delicate molecules without destroying them too easily. This opened the door to studying proteins in much greater detail. Another important breakthrough came from nuclear magnetic resonance, or NMR. This method allowed scientists to analyze the three-dimensional structure of biological molecules in solution. In other words, researchers could study not only what a molecule was, but also how it was shaped. That was extremely important because shape often determines function in biology. This discovery had a huge impact on chemistry and life science. It helped researchers understand proteins, disease mechanisms, and many important biological processes. It also supported progress in medicine, since scientists could study the structure of molecules related to health and disease. I find this discovery very fascinating. It shows that scientific progress is not only about finding new substances, but also about developing better ways to see what already exists. It also reminds us that understanding life requires both identification and structure. In the future, these methods may continue to support drug development, medical diagnosis, and biotechnology. Even though this topic may seem advanced, it gives us an important message. When we learn how to observe the hidden world more clearly, we can greatly deepen our understanding of life. Thank you.
Good afternoon, everyone. Today, I would like to talk about the 2002 Nobel Prize in Physiology or Medicine. I chose this topic because it shows how our bodies are not formed randomly, but are carefully shaped through precise biological control. The prize was awarded for discoveries concerning genetic regulation of organ development and programmed cell death. At first, this may sound technical, but the basic idea is actually very clear. Our bodies are made of an enormous number of cells. These cells do not simply increase one by one without order. Scientists discovered genes that control how organs develop and how cells are instructed to die at the proper time. In order to create a healthy body, some cells must grow, some must change their roles, and some must disappear at the right time. This process is especially important during development. For example, when a baby grows in the womb, organs such as the heart, brain, and muscles must form in the correct places. At the same time, unnecessary cells must be removed. If all cells remained alive forever, the body could not develop into the right shape. This is where programmed cell death becomes important. Programmed cell death, also called apoptosis, is a process in which cells destroy themselves in a controlled way. It is not an accident. It is a carefully regulated system that helps the body maintain order. A simple example is the formation of fingers. In early development, the hand is shaped more like a paddle. Then, cells between the future fingers are removed through programmed cell death. As a result, separate fingers are formed. This discovery was very important. It showed that normal development depends not only on cell growth, but also on the controlled elimination of cells. It also helps scientists understand what can happen when this regulation fails. Problems in this system may lead to developmental disorders, cancer, or degenerative diseases. I find this discovery very meaningful. It shows that life is built not only by adding, but also by removing. It also teaches us that order in biology depends on timing, balance, and control. In the future, this knowledge may lead to better treatments for cancer and other diseases related to abnormal cell survival or cell loss. Even though this topic may seem complex, it gives us an important message. Healthy life depends not only on growth, but also on knowing when something must end. Thank you.